trying to identify the mechanisms that control both neuronal migration, the ability of neurons to reach their final position at the right time, and also the ability of neurons to form the appropriate connections with other neurons. And that includes the formation of nerves, basically, in the developing brain, i.e. axons and dendrites. So in the next issue of Cell, on September 4th issue of Cell, we published a paper where we identify a gene called SRGAP2 that seems to control both neuronal migration and the ability of neurons to differentiate. The reason why we started studying this gene, SRGAP2, is mostly because a member of this family is mutated in severe mental retardation, and virtually nothing is known about the actual function of this gene during brain development. The second thing that triggered our interest in studying SRGAP2, this gene, are its areal organization. The protein contains three areas or three functional domains that had poorly characterized function. One of these domains is called an F-bar domain, and it has the unique property of being able to deform membranes, cellular membranes. One of the deformation that this F-bar domain can induce is the formation of finger-like protrusions coming out of the cell called phytopodia. It turns out that phytopodia are very important for to control both neuronal migration and axon and dendrite branching, the ability of axons and dendrite to branch, which is really a very important prerequisite step, basically, in the formation of connections in the brain. A member of this SRGAP family of genes is mutated in a severe form of mental retardation, referred to as the 3P- syndrome. But the actual mechanism whereby this gene can disrupt brain function is really poorly understood. So our work now that we have characterized that this family of genes can control phytopodia formation, concentrates on the potential function of this gene in spine formation. Spines are basically small protrusions, small phytopodial-like protrusions that are the sites of synaptic contact on neurons. And we have some preliminary results suggesting that this gene family, this SRGAP gene family, actually controls and regulates spine formation. So thereby, this could have direct relevance to explain why, when mutated, these genes can cause mental retardation.